Speaker this morning will be Yves Lespinasse, and as you know, many of you, that we also claim that we in Isofruit project want to increase the quality of fruit. So can this be done, Yves?
How can genetics bring new interesting characteristics to fruit varieties? This is the work we are undertaking inside Pillar 6. And the main objective of the first part of Pillar 6, which is called Gen Fruit, is to understand the inheritance of the main fruit quality traits. We are doing this work on 3 species, on apple, peach, and apricot. And we apply the results for increasing the efficiency of fruit breeding. In apple, for example, we are studying the texture parameters, and they are assessed by global instrumental measurements and analysis of the cell wall compounds and structure. So, the principle for mapping the genes which are important for fruit quality is to work on a progeny. For example, here we are studying a progeny between these 2 selections. The progeny is 150 seedlings, and we are doing in parallel phenotyping of the seedlings and establishing the genetic map through the molecular markers. This type of work is done at 3 times: at the harvest, 2 months after harvest in storage, after storage, and 4 months after storage. And the phenotyping is undertaken through instrumental measurements by penetrometry, compression, sensory analysis, histological analysis, structural and biochemical analysis on cell wall. And as I said, in parallel, we are doing the genotyping. For the instrumentals, are here a texturometer for compression and penetrometry here. The sensory analysis, we are studying crispness, firmness, juiciness, melting, grainy, fibrous, mealiness, and we assess the traits through a scale from 1 to 5 with 4 experts in 2 groups, and we have 2 apples per sample. Another trait, very important, Lars talked a little bit before, is allergy. As you know, allergy in northwest of Europe is about 2% of the population, which is allergic to apple, but also allergic to other fruit, like pear, cherry. And we know that the cultivars differ, really, the cultivars differ in allergenicity. For example, the Dutch variety, Santana, is marketed as such. It doesn't mean that Santana is completely free of allergens, but is compared to other variety as Golden Delicious, for example, Santana is really a big progress. So in this group working on allergy, we are looking for also which genes cause allergenicity. We are searching also for other low allergenic cultivars. So 60 cultivars were tested by skin prick test, and the best 4 cultivars are tested by oral provocations. So, as I said, Golden Delicious is are reference for high allergenicity, and the results show that another cultivar, Elise, is really interesting in this concern. Now, our work is also devoted to cell wall studies for evaluating the texture parameters. So we are looking at, we are doing work on histology, about the cell size distribution and organization. We are also doing works on chemical characteristics, controlling cell wall rigidity and adhesion as sugar composition, fine structure of hemicellulose, which are known to be involved in wall rigidity. This type of work has an application to screen the most interesting variety through the cell wall characterization on apple and apricot collections. And as I said before, at the same time, we have to identify the genetic markers which are associated with these variations.